Hi everyone, this is Dr. Juarez, and I have prepared a shorter version of the lecture than the slides that I provided to you. And that's mainly done just to keep this in a short enough format that you guys will actually listen to it. But also there's a lot of slides on there that were sort of review slides. So I'm going to let you go back through and review those slides, and I'm just going to focus on the things that I think we really need to talk about for today. So today we're going to talk about DNA, we're going to talk about genetic structure, we're going to talk about mutation, a little bit about Hardy-Weinberg, and the bookkeeping of population genetics, and we're also going to talk about protein synthesis. Okay, quick review. DNA and RNA are the molecules that store all of our genetic material. DNA is a double-stranded molecule with a pentose sugar. Deoxyribonucleic acid is what DNA stands for, and the sugar is deoxyribose. DNA structurally differs from RNA in a variety of ways. It's a double-stranded molecule. It has different bases that pair together. And it also has an oxygen at the it does not have an oxygen at the 2' position on the sugar. So if you look here at the bottom, you can see that here is the 2' position and there is no oxygen there. The bases of DNA are adenine, thymine, cytosine, and guanine, and they are listed as they pair with each other. For RNA, similar, we have a pentose sugar, but this sugar is ribose. The bases are different. RNA is a single-stranded molecule, and thymine is replaced by uracil in this particular molecule. And when we start talking about protein synthesis, we're going to see that RNA has two really important roles in the formation of proteins. One of those roles is as messenger RNA, which is the RNA molecule that goes into the nucleus and retrieves the genetic information that's going to code for proteins. And then transfer RNA is the molecule that lives in the cytoplasm and brings anticodons to the ribosome to help build proteins. Okay, so in your textbook, they do a lot of discussion about locus, and locus is the same. It's the same thing as a gene. So it's an interchangeable term for a gene. A gene is simply a sequence of DNA that codes for something. Most often it codes for proteins, but it can code for all kinds of things. When we talk about DNA, or we talk about, genes, we have structural genes and we have regulatory genes. Structural genes are also known as exons, and these code for the production of something. Could be a protein, for example. Regulatory genes are the genes that turn on and off structural genes. Anytime where we have a variation in a DNA sequence, this is at a locus, this is called a polymorphism. So anytime we have a variation in a gene, a DNA sequence in a population, this is called polymorphism. Poly means many, morphism means form. So you're probably familiar with this. You've probably seen genetic material in this format. This is called a karyotype. A couple things, let me just populate the screen with this. So things that we want to remember about human genetic material. We have 22 pairs of autosomal chromosomes, 1 pair of sex chromosomes. Those sex chromosomes come in either an X or a Y, and as they pair up, you get an XY or an XX. We have 46 chromosomes, so we have 23 homologous pairs of chromosomes for a total of 3 billion base pairs and roughly about 25,000 genes. Human autosomal chromosomes are what we call diploid, and anytime you hear ploid, what we're talking about is the complement of chromosomes. So cells that have pairs of homologous chromosomes those cells are diploid. So 23 pairs of homologous chromosomes, those are diploid. The cells that we make in terms of our gametes, those are not diploid because they have single strand of chromosomes. So sperm and eggs have half the complement of autosomal cells because they are going to match up together and they're going to make a human being that is going to be, those two complements of chromosomes are gonna match together. Okay, so before we jump into protein synthesis, let's quickly visit the genetic code and talk about codons. So we have 64 possible combinations of codons for the 20 amino acids. That means there's replication. And that helps us because that means that we have a chance to have some room to protect against mutation. And so in the next slide, we're going to talk about synonymous and non-synonymous changes into DNA with mutations. But because we have several opportunities to code for a single amino acid, this gives us a little bit of protection against things like point mutation. We also have, of course, start codons and stop codons. So this is the synonymous and non-synonymous example here. Synonymous changes basically mean that any changes that occur do not alter the amino acid that is produced. And so that is, again, because of that replication among the codons that we have for single amino acids. So if we look at this particular example of sickle cell, we can see that there was a single point mutation in both cases. But in this particular case on the left, GAA and GAG both code for the same amino acid. But if we look on the right, we could see that GAG and TG do not code for the same amino acid, and therefore that's a non-synonymous change, and the product is a different amino acid, and therefore now we have sickle cell. Okay, so I've provided for you sort of two ways of looking and thinking about protein synthesis. This is a single slide that gives you a kind of a very quick overview of protein synthesis, kind of breaks it down into 3 steps. The first step here is assembling the protein, and that basically what we see is that the DNA sequence is transcribed onto messenger RNA. The molecule is spliced to remove introns and some of the exons. And remember, exons are the coding materials, and introns are the non-coding materials in the genetic code. We used to call introns junk DNA, and the splicing happens to produce kind of sort of like a final or a mature messenger RNA molecule, and then the messenger RNA molecule is then translated into the protein in the cytoplasm at the level of the ribosome. And then you can see here on this particular slide that we have these nice, like, folded up 3-dimensional proteins. Okay, so the next 2 slides break down into about 8 steps what is happening and where it's happening in terms of protein synthesis. So if we just look at this little diagram here, we can see up on the top left corner, we have the nucleus. Inside of there is the DNA. The DNA never leaves the nucleus. But we could see that in the nuclear envelope, we have these little perforations, and the perforations are the way in which messenger RNA goes into the nucleus. DNA only unzips the segment of the genetic material that's going to be transcribed. So messenger RNA lines up along the unzipped portion of DNA. A transcription occurs, which is basically the copying of the mirror image of the genetic blueprint. Messenger RNA leaves the nucleus, goes into the cytoplasm, finds this sort of weird looking gray structure here, which is the ribosome, and docks at the ribosome. There are ribosomes in our cells in several locations, just within the context of the cytoplasm, but also on the rough endoplasmic reticulum. And so messenger RNA will find a ribosome, it will dock at the ribosome, and the anticodons are read. The codons are read, and then the anticodons are going to be brought from the cytoplasm by transfer RNA. So transfer RNA brings those anticodons to the ribosome, and those codons link up into a chain, polypeptide chain. And then the polypeptide chain, once it's completed, it's folded and packaged into the three-dimensional structure of the protein. So why do we care about protein synthesis? Well, we've already seen the example of sickle cell, and we could see that protein synthesis is a place where transcription errors or translation errors can occur. And anytime there is room for mutation, this could be something good or it could be something bad. And so protein synthesis is really critical because protein synthesis is the building blocks of life. And all of our hormones, not just our tissues and our blood, but it's also all of the signaling pathways, our hormone and enzyme pathways that we use to kind of make our bodies work. So it's a good place for us to kind of focus on evolutionary change, and we can also track mutations in proteins and in amino acids. Okay, so a SNP is a single nucleotide polymorphism, and a SNP is just a specific DNA base that varies in the genome among populations. So what we're really talking about is genetic variation, okay, genetic diversity. And so how do we measure that genetic variation or that genetic diversity? We measure what we call nucleotide diversity. So it's these places where we have these base variations. And it turns out that in the history of humans, as I mentioned before, we have had several dramatic population declines. So we've had population bottlenecks that have made the population of humans quite small. And it turns out that small population size is sort of the antithesis of nucleotide diversity. And so every time you have one of these dramatic bottlenecks, you lose quite a bit of your nucleotide diversity, and thus you lose your genetic variation. So if we look down here on the right, we could see that on the bottom we have this scale of nucleotide diversity, which is synonymous with genetic variation. And we could see that nucleotide diversity is indicated by pi. And if we look all the way to the left, we can see that humans have very little genetic diversity in comparison to, or genetic variation in comparison to a lot of the other living animals and creatures on Earth. And again, that has to do with the dramatic population declines that we've seen in human history. So let's quickly talk about how we calculate nucleotide diversity with this little example here. And this is figure 4.5 from your textbook. So we look here on the upper left-hand corner of the screen, and we can see that there are 3 genetic sequences that are 9 base pairs long each, okay? And if we look at the places where we see the black letters, those black letters indicate that there's no difference in terms of the code in those sequences. So if we look at the 9 base pair sequences, we can count that. Let's just count on one sequence. We can see that there are 7 black letters on each of these 3 sequences. So there's 7 on the bottom, 7 in the middle, 7 on the top that are black letters. And so just kind of looking at how we're setting up this calculation, we're comparing the total number of sequences, which is what the number 9, that's in the denominator. Okay, so that's the total number of sequences that you have on one strand. So that's where the 9 comes from. Then what we're doing is we're looking at the similarity and we're looking at the difference. And so 7 of these areas here, 7 of these bases do not have any variation. So at 7 sites, there's no variation among the sequences. And so that's where the 0 comes from. But at 2 of the sites, you can see that we have red letters. So at 2 of the sites, we have bases that are in red text, and that means that the sequences are differing. And if we look at these differences, we see that in 2 out of 3 possible pairwise comparisons, we have difference. So at those particular sites, the difference is 2/3. And so the 2 comes from the 2 sites where we have difference, and then the 2/3 is the amount of the difference that we have. And so that's why we're multiplying the 2 by the 2/3. And if we carry this all through, then we get nucleotide diversity of 0.148. And if we come down here, back down here to this diagram in the lower right-hand corner, we can look at this little measurement of nucleotide diversity on the very bottom of this diagram. And we can see that nucleotide diversity goes from 0 to 1. And so the 0.148 is quite a bit over here. So we're kind of looking at let's see, unicellular eukaryotes and prokaryotes. So that's kind of where we're seeing that level of nucleotide diversity. You can see that humans are way, way, way on the left and very, very limited in the amount of nucleotide diversity that we're seeing and that we're calculating. Okay, so let's quickly shift gears a bit as we start to kind of get into the segment of our lecture that is going to go into mutation. And what we want to do here is we want to conceptualize mutation for what it is. Mutation is a dramatic, it is one of the most powerful ways in which we increase genetic variation into the genome. And so as we transition into thinking about mutation, what we really need to do is we need to transition into thinking about ways in which we introduce variation into the genetic material, into the genome, okay? And so the place where we need to start to think about that, to put our heads into that space, is talking about meiosis. And then we're going to transition from there into mutation, a discussion about that. Okay. So let's put this up here. I am not worrying about you remembering from high school biology all the steps of meiosis, prophase 1, metaphase 1. No, I'm not worrying about that. I want you to focus on the ways in which meiosis increases genetic variation. And this is not simply in humans, although we are going to be talking about a human example. Any sexual reproducing organism is going to use meiosis, which is the creation of the haploid egg and sperm. And so basically we go through a reductive process to make those sex cells. And we basically go through a duplication of the genetic material, and then we go through the double reduction process to basically go from 46 single-stranded chromosomes to in a single cell to 4 daughter cells that each contain 23 single-stranded chromosomes. Okay, so for a quick overview of the whole process, you can look on the left-hand side of the screen, but for right now, what I want you to do is to focus on the right-hand side of the screen, because I'm going to point some things out to you that are really critical for the next couple of slides that we're going to talk about. So just a reminder, when we talk about ploidy, remember, anytime you hear the word ploid or ploidy, like as in haploid or diploid, we're simply talking about the complement of chromosomes that are in a cell. So a haploid cell has 23 single-stranded chromosomes. A diploid cell has double-stranded chromosomes. Okay? So that's the difference. 23 double-stranded chromosomes versus 23 single-stranded chromosomes, half the genetic complement. All right, let's go to this diagram on the right here, and let's focus on some of the important aspects of these stages. What we see happening basically here is the replication of the chromosomes and the way in which they are segregated being kind of put forward in steps. So the red material is the paternal genetic information. The blue material is the maternal genetic information. So remember, you get one set of chromosomes, a single strand of 23 chromosomes from your mom, and a single strand of 23 chromosomes from the dad. When the egg and sperm meet, that's what we're talking about. There's the maternal complement and the paternal complement. Okay? Now, what we're going to see is that in metaphase 1 and in prophase 1 of meiosis, some really critical things happen that affect, that basically create new genetic diversity that didn't previously exist. And so this is why we say that yes, you have the genetic complement, the genetic information rather, from your mom and your dad, but you are something new that never existed before. Okay, so let's talk about what are the 3 things that we're going to discuss in terms of how meiosis and increases genetic variation. So specific to meiosis, we're going to see independent assortment or segregation as one of the ways in which meiosis increases genetic information, making something new that never existed before, and then the process of recombination and what that does to make something new that never existed before. And then sex in and of itself, sexual reproduction, has the concept of random fertilization. Okay, and so basically what that means is that you have 8 million possibilities for the egg and endless possibilities for the sperm, and you never know which sperm and which egg are going to meet together, and therefore fertilization is random because the genetic material that could meet is there's an endless number of possibilities, basically, of what that could be. So let's talk about independent assortment. It's also known as segregation. Okay, and let's talk about how that increases genetic variation. So independent assortment refers to the arrangement of the chromosomes in metaphase 1 at the metaphase plate. And this arrangement determines which chromosomes are going to be then packaged together later on down the line when those single-stranded haploid daughter cells are eventually created. And so there can be a number of combinations with the way in which the chromosomes are arranged during metaphase 1. So this little example kind of explains to you, right? So on the left here, we have a situation where the segregation of the chromosomes created a daughter cell here, which is going to go through additional reduction processes, right? So this is very early on, remember, in the meiosis 1 process. But basically what's happened is that you have a situation where this particular cell has all of the material from the paternal complement. The one next to it has all the material from the maternal complement, but then this one over here has a combination of both maternal and paternal genetic material. And so what happens here is that this process of segregation or independent assortment creates already something new. It creates variation that did not exist previously. So when we think about when we think about segregation, right, and we think about kind of how this works, we necessarily need to think about the way in which the loci on the same chromosomes or on different chromosomes are going to be segregated. Okay, so when we talk about the concept of linkage disequilibrium, basically what we have is that we have a situation where usually because they're close by, these alleles are located close on a chromosome, they're always going to be segregated together. And so when this happens, genetic variation goes down because the linkage disequilibrium basically causes these to always segregate together. One of the ways that we can break this up and we can increase that genetic variation is with recombination. And so recombination is basically this process, also known as crossing over, that happens in prophase 1 of meiosis. And so basically what happens is that you have homologous chromosomes that will actually lay on top of each other, and you could think about the genes on the chromosome like beads on a string, and these chromosomes basically when they touch, they essentially exchange material. And so a homologous chromosome is a chromosome that codes for the same thing. So you're only supposed to exchange material with other homologous regions, right? And so that is one of the ways in which you can get rid of linkage disequilibrium, because you can go in and you can kind of exchange this material, and so you can, quote, unlink these closely these traits that are close on the chromosome. And so therefore, not only are you creating new variation with this whole concept of crossing over or recombination, but you can also get rid of some of those problems of linkage disequilibrium. So you can prevent traits becoming linked and constantly segregating together, and therefore these common combinations, like, for example, I mean, this one's very complicated, but I'll just give you an example that you can kind of relate to: freckles and red hair, right? You can unlink some of these traits from segregating together through recombination. So recombination does two things. It creates new variation that never existed, and it also supports variation by potentially unlinking traits that have become linked and are in linkage disequilibrium. Okay, so we're going to shift a bit to talking about mutations here in a second, but I want to talk about random fertilization first. And because we're going to be talking about random fertilization, we need to remind ourselves about Hardy-Weinberg and some of the formulas that are important for Hardy-Weinberg, and kind of the difference between allele frequencies and genotype frequencies. And there's much more to say about this, but I want to help you to understand kind of some of the impacts about random fertilization and how random fertilization not only increases variation, but also impacts offspring. Okay, so just as a reminder, Hardy-Weinberg, which is the bookkeeping of population genetics, we use two formulas to do the bookkeeping of population genetics. The formula at the top, p squared plus 2pq plus q squared equals to 1, is a formula that's really talking about populations. So in this particular formula, we're interested in individuals and we're interested in their genotypes. And so I'm going to use the mice here as our population, and we're going to talk about what each of these terms mean. So when we see the term p squared, the term p squared refers to the homozygous dominant individuals in the population. So that means that these are the individuals in a population that have 2 copies of the dominant allele. Okay, so in our little population here, you can see that our mice have 2 alleles. So that's a genotype. They have 2 alleles that are showing. And in this particular population, what I'm going to say is that the mice that have the allele A sub 1, those mice represent our homozygous dominant alleles, our homozygous dominant genotypes. Okay, and so those individuals are represented by p squared. 2pq represents the individuals in the population that have heterozygous genotypes. And so this means that these individuals have 1 copy of the dominant allele and 1 copy of the recessive allele, okay, in their genotype. And so that's going to be represented by the green mice in the middle here. So you can see that they have A sub 1 and they have A sub 2. So remember, we're doing bookkeeping right now. And so we are delineating what these terms mean, and then we're going to basically like figure out how we're going to count these individuals up, okay? So heterozygotes are indicated by 2pq in our population. Now, individuals in our particular population that have A sub 2, those individuals have a recessive genotype, and those individuals are going to be demarcated by q squared. So whenever we see this formula here, p squared plus 2pq plus q squared, the couple things that we want to think about are we are talking about individuals in a population, and we are talking about the frequency or the percentages of those specific genotypes in the context of the population. Okay? Now, when we come down here to the second formula, P plus Q equals 1, we are no longer talking about individuals. We're only talking about allele frequencies. How many P's are there? So in this case, P represents A sub 1. So how many A sub 1s are there in our population? How many A sub 2s are there in our population? What's the total number of each in terms of the percentage, right? And so that we know that in our equation here, P plus Q equals 1. So what that basically means is the percentage of the dominant alleles, or the frequency of the dominant alleles, plus the frequency of the recessive alleles equals to 1. Okay, so those are the things that we need to remember for our next slide. Okay, let's talk about random fertilization. And the reason why we needed to talk about Hardy-Weinberg is because we can see here that we now have, right, information on various populations. So we have the adults, that's the parental population, and we have the offspring, right? That's the second population, and we have the Punnett square here. We've got all of these genetic frequencies, so that's why we needed to kind of introduce the concept of Hardy-Weinberg back into the mix. Okay, so Hardy-Weinberg, our bookkeeping method, makes several assumptions in order for us to use the methods of Hardy-Weinberg to do the bookkeeping of population genetics. One of the assumptions that is made, there are 5 assumptions. One of the assumptions that is made is that evolution is not occurring, okay? And so when a population is in equilibrium, basically what is happening there is that change is not happening. So there's not any change in genetic frequency or in the allelic frequency over time. So evolution is not happening when a population is in Hardy-Weinberg equilibrium. Okay? Now, a population can become, for a particular allele, they can get into Hardy-Weinberg equilibrium for that particular allele because an allele can become fixed in a population, right? And so what this is here on letter B, I'm giving you an example of when a population is in Hardy-Weinberg equilibrium, and these are frequencies, okay? So we have reached an equilibrium state when we have basically 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Okay, and so when we're reaching a state like this, an example like this of Hardy-Weinberg equilibrium, what we start to find is that changes in the genotype are no longer possible unless we introduce mutation. So it's a situation that we're going to talk about in a few slides, but basically what happens is that for a specific allele, we can have that allele become fixed, and then with, mutation and natural selection, something new can be introduced, and this particular allele can go through positive selection and move through a population. Okay, so let's kind of look at what's going on with this parental population and this offspring population. So I want everybody to look up here on the top left with the parental population. And so what we're seeing here down at the bottom, we can see that we have the A sub 1, so that's going to be in our particular example, that's going to be our homozygous dominant individuals. And then we have our A sub 1, A sub 2, those are our heterozygote individuals. And then we have our A sub 2, those are our homozygous recessive alleles. And you can see that on top of each of these blue lines, we have the frequency. So you'll also notice that in this adult parental population, we don't have any heterozygotes. And so this particular population is, out of Hardy-Weinberg equilibrium. And we can see the gametes that are produced in terms of the specific alleles themselves. So let's just kind of remind each other. In this first graph on the left, what we're seeing is the results of p squared plus 2pq plus q squared. But as we go to the gametes, what we're seeing is P plus Q equals 1, because now we're just talking about the frequency of those alleles. We're not talking about individuals, right? So the frequency of the A1 alleles is 0.3 or 30%, and the frequency of the A2 alleles is 0.7 or 70%. And when the parental population mates with each other and they actually produce offspring, we can use our Hardy-Weinberg information and our Punnett square to understand what the possibilities of the offspring are. So we could see the eggs are over here on the left-hand side, the sperms are over here on the top. And we can see, okay, we have a situation where we have in the A's, we have A1, 0.3% and 0.3% on the top, right? And then down at the bottom, we have the A2, and of course on the right on the top, we have the A2. And if we cross these to make our allele possibilities, we can see that we get the creation of heterozygotes, right? And so even though the parental population was out of Hardy-Weinberg equilibrium, we get the creation of those heterozygotes back into the population because of random fertilization. So random fertilization increases the genetic variation and increases in this particular case, the heterozygotes in that population, because the Punnett square represents that random fertilization, right? We don't know what combinations are gonna come up, but if we kind of put the calculation out there, we can see that these are the possibilities of the 4 opportunities that we have. And we can see that in large part, right, we get this creation of this heterozygote population that we didn't have in the parental population. And you might be thinking like, okay, well, why didn't the parents have any heterozygotes? Well, there could have been a cataclysmic event and all those people, those individuals got killed, and that's why we didn't have them. And so there's lots of reasons for it, but it could have been something like that. So another interesting fact here is that even though within the second generation, the population goes back into Hardy-Weinberg equilibrium, we could see that if we break it down and go into the allelic frequencies, even though we've got the creation of heterozygotes, evolution did not occur because we went back to basically the same exact percentage of the alleles, 0.3 and 0.7, just the distribution of them changed. So it wasn't a change in gene frequency. So when populations go out of equilibrium, that is maybe an indication that evolution can occur. It didn't happen in this particular case, but it could happen. When things are not in equilibrium, that is a place for variation to kind of slip in. And of course, random fertilization definitely impacts and increases variation. Okay, so let's talk about mutation. So mutation is one of the most important ways in which we introduce variation into the genetic material. Now, your book covers 5 types of mutations that alter chromosome structure. There are many, many, many other types of mutations. And we're going to cover the ones that are covered in your textbook. I've added 2 more that are not necessarily discussed in depth. But what I want to say is that the main mutations that we care about are either mutations that alter chromosome structure, which is what is covered here on this slide, deletion, duplication, inversion, fission, and fusion. But we also care about mutations that alter the number of chromosomes, okay? And so chromosome number can change, and this leads to things like trisomy, trisomy 21, trisomy 20. There's lots of versions of trisomy, or monosomy. And so basically you can have 1 strand chromosome instead of the multiple strands that you need, or you could have 3 chromosomes instead of 2. And so this can cause, the change in number can cause serious conditions. Some of them are deadly. Some of them result in obviously in some kind of a disease that can have cognitive impacts. Okay, so briefly, deletion is pretty self-explanatory. A segment of the chromosome is deleted. Duplication, a segment of the chromosome is duplicated. Inversion, a segment of the chromosome is switched, like it's inverted and then reattached so that the information is out of order. With fission, we have a chromosome being spliced into two, and with fusion, we have a chromosome being added to another segment. So they basically combine when they're not supposed to. Couple other things that are not necessarily, I guess they're covered under the fission and fusion, but basically with translocation, what you get, and you can see kind of here's the arrow here on the karyotype, and then down here at the bottom on the karyotype is basically material is swapped with another chromosome that's not a homologous chromosome. And this is not a good mutation. There's not any, as far as I know, there's no good outcomes for this kind of thing. So this is translocation, chromosomal translocation is one of the major forms of childhood cancer. So I'm sure you've heard of these like babies being born with cancer and you're like, how is that possible? Here you go. Chromosomal translocation, that's how it can happen. Non-disjunction impacts the number of chromosomes, as I mentioned, and so basically what happens is that during meiosis, the chromosomes fail to separate. So in the daughter cells, those 4 daughter cells that were produced at the very end of the meiosis process, we're supposed to have a single strand, right? A single chromosomal strand in there, but instead you have multiple. And so now you'll get individuals that have multiple chromosomes and at a single location, and this is not good. So that happens as a result of non-disjunction. Okay, so let's talk about mutation rates and mutation in general. So every time a person makes an egg or a sperm, roughly 1 out of 108 of the DNA bases has a new mutation. And essentially, mutation is basically one of the key ways in which we introduce genetic variation into the genome. Okay? So, when we think about mutation, we often think about mutation being negative. And it turns out that that is often the case. Any kind of mutation that affects fitness, more often than not, those mutations are negative. If we think about the rate of mutation, the rate of mutation is different in different groups of organisms. And so species that have larger genomes tend to have higher mutation rates. And so if we look at this the little kind of graph, it turns out that things like bacteria and some of these little kind of single-celled organisms tend to have quite different mutation rates than mammals and invertebrates and plants. Plants have a very large genome in some instances in comparison to humans, and so they can have many more mutations than what we see. But in any case, mutation rates vary in different groups of organisms. Let's see here. What else did I want to tell you about this? Oh, pleiotropy. In pleiotropy refers to conditions where a single mutation can have a very serious impact on multiple sites. So examples of pleiotropic conditions are things like achondroplastic dwarfism or albinism, where you have a single mutation that can then have a variety of different effects on different organ systems and affect different, a whole bunch of different traits. So that's an example of pleiotropy. Okay, this is not something that I'm going to talk too much about. I just wanted to put this up here for you to review yourself. We did talk a little bit about epigenetic inheritance. I'm going to touch on it again later on in the semester, and we're going to read some really cool stuff about it. But essentially, epigenetic inheritance happens, one of the main ways in which it happens is through methylation. And so you can get a situation, remember I told you, the genetic code is like a keyboard. And your genetic code exists in many different environments. So I think I gave you the example of the womb being an environment. And basically what can happen is that these various environments in which we exist throughout our lives can alter gene expression. And one of the ways that that happens, one of the mechanisms for that is through methylation of our bases. And so the C and A bases of DNA can be methylated, and this can alter how specific genes are expressed. And if this epigenetic methylation, if this methylation happens before you have offspring, it's heritable. So it's something that can pass on and can completely alter the ways in which genes are expressed. Maternal effects have to basically do with the phenotype of the mother having an impact on the offspring, and while they will have an impact on that direct generation, these don't have, they don't contribute to long-term evolutionary change. And then cultural inheritance, basically you transmit the knowledge of certain behaviors through complex cultural practices, and it impacts learning and what you're able to do. And then this is kept through the generations. And this could be something that contributes to epigenetic inheritance, creating new environments where methylation occurs or doesn't. So methylation tends to be negative and tends to have negative consequences like aging and disease consequences. But we're learning more about that every day. And so I think that there's a role for cultural inheritance in this particular discussion. Okay, so now we're going to start getting into a little bit more of the Hardy-Weinberg talk. And we're going to be talking about specific mutations, okay? So in order to do that, let's kind of lay the groundwork here with some of the things, that we need to know about. Absolute fitness and relative fitness. So absolute is the simple count of the number of zygotes that you produce over your lifetime. Relative fitness talks about absolute fitness divided by the fitness of the reference population. So this is the population average. And relative fitness is the more commonly used term here. Positive selection, there are many different types of selection, but positive selection is when an allele that positively affects fitness is being selected for by natural selection. There's also negative selection, and there's also balancing selection. And we could talk about those more later, but I'm going to give you some examples of positive selection, and I'm going to talk to you about the selection coefficient and how we calculate that. And we're going to see an example in a second here. So the rate of evolution at a locus is indicated by the change of P. And so basically, this is the change over time of a specific allelic frequency. And so you can see here, P is allele 1, and 1 minus P is allele 2. You could also think of this particular example in the terms of Hardy-Weinberg. So P in that particular example would be P, and then 1 minus P would be Q. Okay? Okay, so let's talk about positive selection and calculating the selection coefficient. So what we have here, I want everybody to look at the diagram at the top. Okay, so the first diagram that we have at the left, at the top, is a population of juvenile mice. Okay, and then as the juvenile mice grow, we can see that the surviving adult population, some of the individuals did not make it to adulthood. And so the death of some of those has affected the frequency of alleles in the population. Okay, so when we see P is going to be the frequency of A sub 1 alleles in the population. Okay, so all of the turquoise rats have A sub 1. So they are A sub 1 homozygous rats. Okay, then if we look at this, and this we're looking here at this population on the left. Okay, so the juveniles that are starting out. So we have 4 turquoise rats that have A sub 1 as their alleles for this particular genotype. Okay, then we could see that we have 2, 4, 6, 8 heterozygotes. So we have 8, and those individuals have A sub 1, A sub 2. And then we have 4 homozygotes of A sub 2. Okay? So now what we're going to see is we're going to have a situation where A sub 1 was fixed in the population. So in this population of juveniles, A sub 1 was fixed in the population. But then we have a situation where the environment changed. The environmental context has changed. And now A sub 2 is going to be positively selected for. Okay, so you can see the surviving adults is after that climactic or that environmental change occurred. And we can see that environmental change that killed off some of these rats was negatively impacting individuals that had the A sub 1 genotype, right? So we could see that whatever happened in the environment here, having the A sub 2 genotype was more beneficial, okay? And so the surviving population of adults reproduces, and then we can see that we've got that change in the genotype frequency, right? Where we have A sub 2 being favored. And so the result here, of course, is that we have an additional individual that is homozygous for A sub 2, A sub 2. Okay, so let's talk about how we discuss positive selection in terms of selection coefficients and in terms of sort of the bookkeeping of Hardy-Weinberg. Okay, so I put some of this down here for you. Remember that when we talk about populations, we're going to use this formula, p squared plus 2pq plus q squared equals to 1. Okay, G1 represents the first population of juveniles. Okay, so this population up here on the left. In that original population, we had 0.25 p squared. We had 0.5 heterozygote individuals, and we had 0.25 homozygous recessive individuals. Okay, so we had a population that was at equilibrium, and this population had become fixed. And in that particular population, A1 had become fixed. And then we had that cataclysmic environmental situation. And then we had a situation where whatever that environmental change was, it allowed natural selection to act, and it created a scenario in which the A2 variant was more beneficial. And so when we go into that offspring population and we put the frequencies there, right, of the individuals, we can see that we've got a change, right? So we've got in terms of individuals, this doesn't necessarily mean evolution has occurred because we can only see that on the level of allelic frequency over time. But in terms of evolution, we have a different percentage now of p squared, and same percentage of heterozygotes, but a different percentage of q squared. Okay, so let's look at the change in the allelic frequencies themselves. So A1 plus A2 represents the allelic frequencies, right? We've got in the first example, in the first population, in the G1 example, we have 0.5 and 0.5. But in the second population example, we have 0.42 and 0.58 as the frequencies of the alleles over a single generation. Okay, but still a change in gene frequency over time. It's a single generation. Now, we can use this formula here. The change in P is roughly equal to the selection coefficient plus P times 1 minus P. So remember, this is basically the same thing as saying the selection coefficient is times P and then multiplied by Q. Okay, so if we look down here on the bottom left, I've calculated it out for you so you can kind of see how this works. So in order for us to calculate this out, we need to have at least 2 generations. So we need to have the parental generation and then we need to have offspring generation so that we can understand what the change in allelic frequencies is. Okay, so in this particular instance, right, we're looking at, we're equating, right, P with the A2 because that is the allele that's being positively selected for. Okay, so let's look at this allele and we'll say, okay, in positive selection, what is the selection coefficient for this allele? So the change in P for the A2 allele is basically 0.08. And you get that from subtracting 0.5 from 0.58. So that's what goes over here for delta P. And then we have S, which is our unknown. And then we have P, which again is the allele that we're selecting for in our final population, which is 0.58. That's the frequency there. And then 1 minus P is the A1 allele. So that's 0.42. And if we multiply that all together and solve for S, we find out that S is 0.33. Okay? And so this is the selection coefficient. Now, let's translate this in terms of relative fitness. And then on the next slide, we're going to see what this all means. Okay? So in order for us to calculate relative fitness is simply W, which is the relative fitness representation, equals 1 minus S. And so 1 minus S is 0.67. So the relative fitness of all the individuals that are going to have the A2, right? This is going to be the relative fitness of this particular population. So let's look at what that means for the impact of positive selection in this particular scenario and the selection coefficient and how that impacts how an allele gets fixed in a population. So in positive selection, okay, this is only in positive selection because negative selection, the selection coefficient can be negative. So in positive selection, the higher the selection coefficient is for the allele that is benefiting fitness, the faster that allele can be fixed in the population. Okay, so the way that we calculate how long it's going to take for that allele in terms of generations to be fixed in the population is we simply divide 4 by S. So in this particular example here, the S is 0.05. And so if we divide 4 by 0.05, then we see that it's going to take 88 generations for something that has a selection coefficient that's being positively selected for and has a selection coefficient of 0.05 to be able to become fixed in the population. Now, in the example that we had on the previous slide, our selection coefficient was 0.33, so much higher. So it would only take 12 generations for this particular allele to become fixed in the population. Okay, so for things that are being positively selected for, the higher the S, the higher the selection coefficient is, the faster it is going to become fixed and spread into the population. Okay, so when we're talking about alleles, we always have to keep in mind that these genotypes are going to code for phenotypes, and these phenotypes are going to have trade-offs. And so this is another figure from your textbook, and it's really kind of focusing on this whole issue of genetic trade-offs in survival and in mating success. And so the individual sheep at the top, they have normal horns, and they have high mating success because they do competition for females, right? And they do this through headbutting each other and having these giant battles. And so they have high mating success, but they have low survival because they get killed off. But the other phenotype that is possible in these types of sheep, because of this sort of dramatic polymorphism that they have in their genomes, is that they can have these really tiny horns. So the guys at the bottom have these really, really small horns. And they have pretty low mating success as a result of these really small horns. They're called vestigial horns. But they have high rates of survival because they're not banging each other's heads against each other and fighting for females. And we bring this up because, very similar to sickle cell anemia, what often happens in this situation is that because you have this trade-off where the individuals with the normal horns have high mating success, but they have low survivability, and then the individuals with the vestigial horns have low mating success but high survivability, right? What happens is that you get a selection for heterozygotes. And so both of these alleles are maintained in the population. Okay, and so that is called overdominance. That's a process called overdominance, when basically you have a situation where there could be something that is slightly deleterious, or where in this particular case where the mating success is low, but basically there's a counterbalance to it, usually with survivability, and as a result of that we get the heterozygotes being selected for. The same is true in areas where malaria is high. We have there's a benefit to being a carrier for the malarial gene, but there's also not a benefit to being an individual that has sickle cell, but we see that that particular gene is maintained in the heterozygotes because it offers protection against malaria, right? So similar trade-off. Okay, so we sort of alluded to this a little bit when we talked about linkage, but essentially what happens is that as beneficial mutations are selected for, and as they spread to fixation in populations, you can have neutral mutations that don't have any effect. And as long as those neutral mutations are on sites nearby the beneficial mutation on the chromosome, then what can happen is that these neutral mutations can also become fixed, on the chromosome. And as the beneficial mutation spreads into higher frequency, this neutral mutation that has no effect acts as a hitchhiker, and it also becomes fixed. And the effect of this is that once the population, or once the beneficial mutation becomes fixed, we start to see a lack of genetic diversity. And that whole process is called selective sweep. Okay, so this is our very last slide here, and I just want to go over a couple of things that happen in terms of the allelic frequencies and the trajectories under different types of selection. So we talked about positive selection, but there are other things that can happen. Overdominance and underdominance. So with positive selection, remember that what's happening here is that natural selection is favoring an allele that offers a benefit or increases fitness. And so basically what happens is that that particular allele is selected for, and that that particular allele that benefits survival is selected for. The higher the selection coefficient, the faster that allele is going to be come fixed in the population. We talked about overdominance, the situation with the sheep, where we have a situation where there is an unequal benefit. There's some kind of inequality between the outcomes. And in these particular situations, we see heterozygotes being selected for. And then we can have another situation called underdominance, which is at the bottom here, in which case heterozygotes are selected against. And with underdominance, heterozygotes have the lowest fitness, and so therefore they're being selected against. So I'm going to end here. Like I said, this is a truncated version of your lecture because you can see it's already an hour long. And it's just way too long to cover all the slides. But the slides that were cut out were for the most part review. So please let me know if you have any questions. We will be having class next week. I will be in class on Tuesday. I will be not in class on Thursday. And we will have a pre-recorded, so therefore we will have a pre-recorded lecture to watch on the 15th. Okay? All right, everybody. Thank you. Bye-bye.